Happy to welcome back to Immunology and Biotechnology online by Dr. Hong. And so in this week's lecture or topic, we are first going to look at autoimmune disease from a cellular and molecular standpoint. We don't go into too much into specific diseases because you will see me in the same person in another lecture, in another course about talking about those diseases. Okay, now if you wonder why am I wearing tie and shirt at home, it is because I don't have a lot of street clothes, I realize, and instead of thinking about what to wear for today's video, I just revert back to my normal dress code. Okay, so enough for the smart talk, and let's go to the lecture. Alright guys, welcome to week 2 of the immunology lecture due to this COVID-19 closure. Now this week we're going to have 2 topics for discussion, and the first topic is autoimmune disorders. Okay, now this particular lecture is not going to talk about too much on particular diseases, okay, autoimmune diseases, because we are going to talk about those topics in your GEM module, okay, particularly partly by me again. So I think by the end of next week you're going to get very tired of my voice. I'm pretty sure of that. So before we get tired of my voice, let's get into today's topic. Alright, so today's topic, we're talking about autoimmune, and we're basically looking at most of the cellular mechanisms, so things happening behind the scene, okay, i.e. the B cells and T cells. How are they being dysregulated in a way that they turn against us? Alright, some of the learning objectives, okay, those are again pretty straightforward. I won't read about it on my recording here. You can read those on your own. Now I'm gonna, toward the next part of the video, I'm gonna point out some diseases that are autoimmune related, but are not presented in your PT modules normally. So I'm gonna touch base upon those a little bit to wrap up this topic. Okay. So introductions. Okay, so autoimmune disease basically is your body turns on itself. Okay, it is a term that applies to a very wide variety of diseases. Okay, there has been shown that the prevalence in industrialized nations is approximately 5%. So it's some reason, like allergy, like what we talked about last week, this appeared to be a higher prevalence or incidence in industrialized country compared to other countries that are not so much industrialized. So there's for example here, the thyroiditis, okay, something about your thyroid, okay, inflammations due to autoimmune attack, okay, it's estimated to be about 2% in the US, okay, and also rheumatoid arthritis estimated to be 1 to 3% in the US. Alright, so the CDC NIH projected the prevalence of rheumatoid arthritis as high as 18% by now. Oh no, this is a prediction. I don't have a real record for you, but it's a lot, and then we're seeing a lot more drugs, trying to come on market to help with those type of conditions. So the autoimmune reactions are also, you can classify it as type 2, 3, 4 kind of in a way that make the hypersensitivity, but we're not going to talk specifically in those cases for this topic. Okay, now the root causes, okay, what are the main reasons for autoimmune disease? In collectively speaking, it's a failure of mechanisms of self-tolerance. Okay, now we're going to talk about how B cells and T cells recognizing ourselves, and if you recognize yourself but you cannot tolerate yourself, you started to attack yourself. Okay, so that is the central underlying principles there. So you would have something that are self-reactive, B cell, T cells, and auto antibodies. Okay, antibodies against yourself. Okay. So we're going to first look at T cell tolerance, okay, and then we're going to look at B cell tolerance later. Now remember, we talk about the development of T lymphocytes, we talk about clonal selections and deletions, okay. So these are the principles, general big principles behind T cell tolerance, really. Right, so here there's a term that I want to introduce to you, which is this AIRE, I circled it, which represents for autoimmune regulator, so which is a transcription factor that responsible for transcription of tissue-specific self-peptides in the thymus, and then presenting from the antigen-presenting cells. Now, so you're going to present on the MHC complex or proteins, right? So once these things are being presented, for example, looks like in this picture described here, being presented in the MHC, and there is the T cell interactions. Now, if interaction with a self peptide, okay, here is a self peptide. Now, if the T cell is interacting with this peptide with a very low affinity, okay, that means they do not in this case, they do not receive a signal to switch off a process, and then they will automatically go into apoptosis. So really, in the T cell development, apoptosis, apoptosis, is a natural process. You need something to tell it not to self-destruct. Okay, so if it is too low, okay, that means this T cells are not binding to much of anything, okay, that's kind of useless. Alright, useless things, let's pop it, let's go through apoptosis, let's destroy it, or let's let it die, okay. And then on the other spectrum, if you have a T cell interactions that have very high affinity with self-peptide complex presented by antigen presenting cell, that means they are not good either, so they are very much likely to react with self-peptide and causing problem downward. So, well, then there is a signal going on to let it die as well. Okay, a death signal being sent into it, and then it goes through apoptosis again. Now, only those T cells that have intermediate affinity or interactions with the complex, those it will be able to mature in the thymus and then migrate to the peripheral, and then leaving into the systems and do its job. So you need just like a fine balance between high affinity and low affinity. Okay, medium is good. So in that sense, there are some type of auto-reactivity in a sense, right? You naturally have some recognitions of self-antigens, and in some animal studies, that they're being immunized with certain self-antigen that can lead to autoimmune diseases. So it implies that the presence of cell-reactive B cells and T cells are actually something normal in animal models. Now, coming back to a human standpoint, we have one common example people listed in terms of autoimmune disease is the type 1 diabetes. Okay, in those patients, they may have autoantibody that are attacking insulin, so that they are not they're not able to have enough insulins to do the job. Now, and also there's example being observed, people with multiple sclerosis, they have autoantibody that attack the myelin sheath of the nerve fibers. Okay. Now, however, some of these autoantibodies can be detected in people without any manifestations of these diseases. So, in some way, it suggests that there are some other mechanisms that play a role to maintain tolerance for ourselves. Okay, so during the development process, there are self-recognition, self-reactive, to a small degree, but there must be something else beyond the development, so-called the peripheral mechanisms, that can help with the process to protect us from being attacked by ourselves. Okay, and these peripheral mechanisms is called the inductions of T cell tolerance through ignorance. Okay, simply ignorance. Ignorance is a funny term, ignorant, but in this case, it's implies that the T cell would ignore the antigen, even though people can see it, but they tend to ignore to it. So it depends on the first level of antigens. Okay, if it is below a threshold, it becomes non-responsive. So it's just like people are doing. There's a good example. So if imagine you're in a classroom, imagining you're in a classroom right now, and I'm standing in front of you guys and talking about this topic. If there is a one or two kids behind the back that are, maybe chatting, but if they're not too loud, not disturbing the whole class, it's the level of distractions is not too high, below my threshold, then I will not be responding to it. Basically, I just tend to ignore it. I'm ignorant in that way. Dr. Han simply ignores the distractions. So, now, but if that level of distraction gets high enough, spread across the whole classroom or half of the classroom, that irritates me. Okay, and then I would start to respond to that distractions. So, in a sense, this applies to T cells as well. Now, another mechanism that can induce type of a tolerance is through physical or anatomical barriers. Okay, so if you block off the T cell from accessing certain area of your body, so if there's no access, then there's no response, right? So for example, in the CNS, okay, that is one example. CNS usually have a very low MHC expressions. So remember, T cell need to interact with MHC first, and then if you don't have low MHC, it's not doing anything. Also, some barrier, okay, that can physical actual barrier can block off. Another one that is something we call the BBB, right? Blood-brain barrier is one of those. So other parts of the body, anterior chamber of the eyes, they can also be part that can exclude most T cells from going there as well. So you're blocking access. Alright, so these are and then the third mechanism to induce ignorance is lack of co-activations. So basically, you are not having co-stimulations. You are crippling certain responses. So remember the interactions between the helper T cells. Okay, it needs the CD28 and B cells, interactions as indicated in this picture here. And some tissue cells lack of the lack of the B cell co-stimulators, and therefore you are not able to having a stimulations for those naive T cells activation. So you just lack of something. So you're not giving them another touching point. So you're not touching, you're not activating them. So it is just that one way to induce ignorance. Another final way of doing this is through inhibitions. So there is a marker called this CD152 or CTLA4, stand for cytotoxic T lymphocyte associated protein 4. Okay, CTLA4. Okay, once again, it's cytotoxic. Let me see if I can write it down. Cytotoxic. XIC T lymphocyte associated protein. My handwriting is usually much better than this. Writing on a touchpad, writing pad seems very difficult. Alright, so having this marker, okay, they will compete for B7 binding site. Alright, so you are sticking a a marker in between B7 and CD28. So basically, you're stopping the interactions or co-activation process there. And we have both membrane-bound and soluble form of these CD152. And we have observed that individuals genetically produce less CD152 are more prone to develop certain types of autoimmune diseases and type 1 diabetes, and individuals who can produce more CD152 can are more resistant to autoimmune diseases in general. So another way to induce T cell tolerance is through a deletion mechanism. So if there are absence of co-stimulations, okay, i.e. there are not enough antigens there, there can be inactivated, okay. We'll come back to this term energy in later slides, okay. And then lack of growth factors, okay, you're not feeding them, right, such as IL-2, T cell growth factors, then they're not going to grow, and then eventually they will die. And then through fast induced apoptosis, okay, fast receptors on T cells and with correspond with the ligands on other cells can induce some of the apoptosis process, which leading to T cell death. Alright, continue with the peripheral mechanisms to induce T cells tolerance. You can also suppress, okay, the activations by a new family member here, okay, T regulatory cell, Treg. I think this is our first time to talk about T regulatory cells. So they have CD4 marker in themselves and CD25. So they are CD25, CD4 T regulatory cells. They are unlike other T cell, okay. The Treg cell, the T cell receptors of Treg cell is specific for self antigens, okay. So here is an example here showing you, hey, you have an antigen presenting cell that are interacting with CD4 T cells, with the self peptide, self antigens, and the Treg cells. It's at the same time binding to the same cells, and then perform some regulatory functions where it can interact with and then suppress the autoreactivity of T cells. So here we're in the peripheral, and you have some reactive CD4 T cell that are binding to self-peptide, but we have this Treg cell kind of doing the checking, okay, by releasing the IL-10, which inhibits cytokine synthesis, and also releasing TGF beta, which inhibit the mitosis and promote apoptosis of that self-reactive T cells. It can also produce soluble CD152, which we just talked about, and perform the inhibitions, okay, stuck in between the B7 there to prevent activations there. Alright, here is the summary picture to include everything we talked about for T cell tolerance. Okay, we're still talking about T cells. Okay, remember that. Alright, so this is a normal process where you have antigen presenting cells, and then you interact it and activate the T cells. Now, the first one is you're having an anatomical barrier. This is a immunological ignorance, and basically you block off the process. Okay, physically block off. And then you can, through deletions, through the interactions between fast and fast ligands, okay, you can delete, okay, lead to apoptosis. And you have inhibitions, okay, inhibition, we're talking about having CD152 snuck in between the B7 and then preventing the activation, so no activations. And then we have suppressions where the Treg cells is secreting IL-10, TGF beta, and again lead to no activations. I think this is a nice graph to summarize what I've just talked about in the past 10-15 minutes or so. Okay. Now move on to the B cell branch of the story. Okay, so here it's just a little bit recap. If you want to looking back the B cell lymphocyte development, that should be able to give you a little bit refresh. Okay, now here just a summary, okay, of the central mechanism for selections. Okay, we're not talking about tolerance yet, so this is some of the older type of material. Okay, so remember, there's a positive selections. Okay, basically here through this pre B cell stage where it's checking can B cell produce a functional immunoglobulin receptors. Okay, and in the first stage we are looking at, hey, heavy chain first. If do they have a heavy chains? Okay, if they don't have a heavy chains, they go through the apoptosis process. They are not going to be propagating. Sorry about that, notifications. And then if the and then continue to go through, the light chain gene rearrangements, see if they are producing the light chain. Again, if there's a light chain, it will go through the process into a mature B cells. But if they don't have the light chain, okay, selection for functional light chain. If they don't have it, they go to the apoptosis process. So this is a normal mechanism for selections. Okay. So in terms of the central mechanism for tolerance for B cells, we are actually first looking at the negative selections here. Now, so here is the graphical representations. We have IgM, membrane-bound IgM on the B cells. Okay, now you're asking the B cell, are they self-reactive? Okay, if they are self-reactive, okay, this is screening against those stromal cells. Stromal cells are actually some connective tissue cells that are represented in many places. Here we are in the bone marrow here. Now, if it is reacting with the self-antigens, those guys don't get to leave. Okay, they will be retained. Now, they maybe reprogrammed. Okay, we can correct them. Okay, in a sense. We'll talk about it later. And then if they're not self-reacting, okay, those cells would be able to release into the circulations, and then they would be able to co-express IgD and IgM eventually. So here we're talking about the reprogramming, so-called the receptor editing. So when it first come out, we have something that are self-reactive. It will go through this editing process. In this process, usually what happens first is there is a massive IgM activation and cross-linking with the self-antigen. And at that point, the B cell would stop their development. Okay, they're not going to develop furthermore, and then go through the rearrangement of gene, okay, generating new light chains. And after this new generations, they're gonna go through the presentations again. Now, if it goes through okay, i.e. they're not self-reacting, it will leave. Okay, it will continue to develop, to mature, and then leave the into the circulations. Now, if at that point after the arrangement they're still self-reacting, it will actually go through the apoptosis. Okay, this is beyond hopeful. We cannot reprogram this B cells anymore. Let's let it die by itself. Okay. Now, in terms of the peripheral tolerance, we still have this absence of co-activation. So very similar idea to the T cells, absence of co-development in co-activations. Now, in this case, it's the lack of corresponding CD4 cells that are specific for the same self-antigens as the autoreactive B cells. Okay, remember that's that CD40 and CD40 ligand interactions. Okay, if there is the CD4 T cells, they don't have that same self-antigen that are on there, and basically you will have no CD40L and CD40 receptor co-stimulations, and at that point you are still not able to activate the B cells completely. So here we see the term for B cell energy. Okay, energy means functional inactivation, which is a peripheral mechanisms of B cell selections, basically. Right, so this is the detail of what was happening in between the cross-linking, right, massive IgM activation and cross-linking, in between that step to apoptosis that we just talked about in the last slide. Okay, so after this surface IgM being sequestered, being activated, cross-linked, these self IgM antigens will basically being sequestered in internal cellular compartment. Basically, some of those would be internalized back, okay, so where they cannot reach and react with the antigen, so they become unreactive. Alright, and at that point, a subsequent encounter with a CD4 T cells and the signal sent through the Fas ligand will induce apoptosis in those self-reactive B cells. So the second mechanisms in peripheral selections occurs in the secondary lymphoid tissues, okay, and basically what it means that the immature B cell need to go through the primary follicles and then it reach to a secondary lymphoid tissues. Now those that cannot go through will first become eliminated. Those energetic B cells that cannot go through will also undergo apoptosis. Now after they go through into the secondary lymphoid tissues, there is a factors called the BAFF, B cell activating factor, and in those follicles, those are important for the mature B cells to survive. Now without those, they will not die. So basically this is a way to prevent things that are immature, things that are at the energetic stage to go through and thrive. Okay, you are preventing these problematic B cells entering into your circulations. Alright, we've go through a lot of complicated concepts. Let's stop for a while, look at questions, and recap a little bit. Okay. Alright guys, you're seeing me holding a mic with me right now, and it is time for Who Wants to Be a Millionaire? Fake Millionaire. Again, so by this time, I think all of you are familiar with the process, okay? There is this drop-down menu. It's okay, move your mouse over to the eye icon up there, and you can participate in this round of questionnaire. And we are going to start over from 100 again. So each week, things don't accumulate, okay? It will start from 100 back to 100 again. So let's go to the first 100 questions, okay? $100 questions. Alright, inhibitions, okay, in T-cell tolerance involves, okay, so it's just something that we talked about in the past 15-20 minutes. Alright, so thinking about inhibition. So what if you want to look up your handout, that's okay. Alright, so that is that little graph with 5 categories, okay. Alright, so inhibitions, okay. What does it involve? TNF alpha, B, CD152, C, alright, interferon gamma, and D, anatomical barrier. Alright, so we're talking about inhibitions. Somehow it inhibit the activations, okay. It probably requires some molecules, right? I'll give you 5 seconds to either think of it or look it up. 1, 2, 3, 4, 5. Do you have a final answer? I think you do. Alright, participate. Put a click there, and the final answer is B, CD152. So it is the soluble CD152, then a sneak it in between the B7 there, co-receptors, and basically blocking the initial activation processes. Right? Okay, well now let's go to the second question, which is the $200 questions. Right, so this one is about B cell energy involved. So I asked you one question on T cell, and it's kind of understandably you will see another question that is related to B cells. Okay, now these are some of the newer concepts, so you're welcome to flip through your notes in between. Alright, so it involves IgG activations, IgE activations, IgM activations, and IgA activation. So this is pretty basic understanding of B cell energy. Okay, energy. So think back, look at the graph again. So what type of an antibody initially is on the B cells? Right. So if you think of it that way, it's not that difficult, right? 1, 2, 3, 4, 5. Pull down the menu, put a click. Final answer. C. IgM activations. So that is the antibody that are initially on the B cell during development, and they are being clumped together, okay, and sequestered and needing, going through some type of a reprogramming, right? If it doesn't go well, it will just go to apoptosis. Right, so that is for part 1 of autoimmune, and stay tuned for part 2. Alright.